Good morning. My name is Amy Wong, and my mentor is Dr. Kirsten Limison. Can you imagine not being able to drink your morning coffee, taste your mother's home-cooked meal, or eat your favorite cake for your birthday? Well, these are some of the consequences that head and neck cancer patients face when they undergo radiotherapy. While the purpose of radiotherapy is actually to target and kill the cancer cells, regrettably, the surrounding normal salivary glands are often targeted as collateral damage following radiation treatment. These cancer patients actually face many consequences of oral complications, such as hyposalivation, which is the loss of saliva, mucositis, which is inflammation of your mucosal cavity, and also malnutrition due to their inability to eat and consume food. Now, your salivary glands are actually very important. They help protect against microbial infections, they help you digest your food, and overall, they help maintain your oral health. So you can imagine that dysfunction of salivary glands is actually really problematic for cancer patients. Clinically, there's currently no definitive cure available to restore salivary functions, and these adverse complications actually leads to alteration in their treatment schedule, discontinuation of their cancer therapy, and overall, this leads to a decrease in the patient's quality of life. So my lab is actually interested in identifying the mechanisms that are altered following radiation treatment, and more importantly, what can we do to fix it? So our lab is actually interested in the role of adherence junctions and what happens to them following radiation treatment and during regeneration. So what are adherence junctions and why are they important for salivary secretion? Well, you can think of adherence junctions as mini bridges between two cells. Like bridges, they are able to help maintain structural integrity, which allow those cells to actually interact with each other. They allow for the import and the export of ions and electrolytes, which allow them to help modify and produce saliva. And most importantly, they allow for the signal to go back and forth between these two cells. And this allows the cells to communicate with each other what is happening. So you can imagine that if there's a disruption in these adherence junctions, or if there's a disruption in these mini bridges, this will lead to an alteration in their function and ultimately results in salivary dysfunction. So this is actually what we see following radiation treatment. If we radiate mice, take out their salivary glands, and stain for adherence junctions such as E-cadherin and β-catenin following radiation treatment, we see a disruption of these interactions. Adherence junctions are actually typically found along the membrane of a cell, and they actually help maintain these interactions. However, following radiation treatment, we see that these adherence junctions are actually located in the cytoplasm and in the nucleus, which is the inside of the cell. However, if we gave these mice a compound called insulin-like growth factor 1 or IGF-1, this is actually able to restore these adherence junctions back to the membrane. So future work for my lab and for my project is actually looking at the role of these adherence junctions following radiation treatment and after insulin growth factor 1, which is the regeneration model. If we are able to actually restore these adherence junctions, we are able to bridge the link between these two cells, then perhaps we can find a drug that can actually promote restoration of salivary function. Hopefully in the future, head and neck cancer patients will not have to worry about the effects of radiotherapy, but rather focus on how to treat their cancers and actually regain their health from these cancer treatments. They will be able to drink their morning coffee, taste their mother's home-cooked meal, and enjoy their birthday cakes. Thank you very much.